And then hopefully that will exist so people can use it. All right. And then I share screen. Hopefully it looks good. Both computers. Okay. Um, I don't. Okay, cool. It was showing presenter mode on Zoom, but I think now it should be good. I'll keep that little. All right. So welcome all. Um, like last time, I'm Justin from I'm a six-year grad student in Charles Ariglou's lab. And we talked last time about some RNA-seq data analysis, and today we're talking about what I call epigenomic DNA-seq. I'll talk a bit about which particular techniques, because there's several techniques in epigenetics that are kind of all similar, and then there are some that are quite distinct that I won't talk about today. But to just get started, I'll remind you of last time we talked about RNA sequencing, and the major steps are to isolate RNA from the samples, fragment it into short segments, and then convert those into cDNA. So you have these little pieces of DNA that are going to actually be sequenced. You add some sort of sequencing adapter so that it can be compatible with sort of the sequencing chemistry in the machine, and then perform next generation sequencing and then map all of these small reads of DNA sequence to the genome to figure out what RNAs they came from. And with sort of this DNA sequencing family I'm talking about today, it's basically the same process. So originally we're isolating DNA instead of RNA. We're going to, in some way, fragment the DNA into short segments. That's one way that the techniques vary a bit. You don't need to do this third step of converting to DNA because you started with DNA, so that is a bit more convenient. And then ligating sequencing adapters is still required, and then this next generation sequencing and mapping to the genome is still similar, but done a little bit differently. So to start, sort of the classic technique in this family is chromatin immunoprecipitation sequencing, or ChIP-seq. And this is what a lot of the methods were developed for and still what a lot of people use. So for this, you first need to split the DNA into different pieces. This is often done through restriction enzyme digestion, like I show here, but can also be done through things more simple like sonication or something like that. But in this case, since the DNA is going to be bound to proteins, here I'm showing histones, but this could also be a transcription factor or a chromatin remodeler or anything like that. You have the DNA, but you're going to also have in those small sort of pieces the attached proteins. And using an antibody against a particular target of interest, you then perform an immunoprecipitation. For chromatin, this is usually done to histone modifications, but can also be done for transcription factors and things like that. And so when you do that immunoprecipitation, like this antibody I'm showing here in the corner, you pull down not only that protein, but also the DNA that's attached to it, and you leave these other nucleosomes behind, so the DNA and the histones they're wrapped around. So then once you make this library of DNA, I keep it colored orange here because then that would only be pieces of DNA that were wrapped around whatever protein you were particularly interested in. In this case, it would be a monomethylation mark on this histone. You then still need to do that final step of adding the sequencing adapters and then the actual sequencing. And then instead of aligning to the exons, like we talked about with RNA sequencing, the DNA can come from really anywhere in the genome. It's not going to be exclusively at exons. And you're just going to kind of more simply align it to the genome itself without having to worry about where the exons are or splicing or any of the things we talked about with the RNA. Um, and then something we'll talk about a bit later is that once you have all of these sequencing reads aligned to the genome, you might notice certain locations where there are a lot more than others, and you want to identify where those are. And there's some sort of computational ways to identify those. A newer technique that's pretty much completely replaced ChIP-seq in how people should use it at least, is called cut and run. We'll show actual plots a bit later about how the two compare. But with cut and run, you basically have these magnetic beads that bind to the cell or nucleus of interest. Doesn't really matter what your input is here, whether it's cells or nuclei. You then add an antibody that binds to whatever thing of interest. Here again, I'm using the histone as an example. There's a sort of secondary antibody. Well, not really secondary. It's a P imminase. So this has the protein A that will bind to the antibody itself. And then you make this complex where you have your antibody and this imminase, which is that same restriction enzyme that we had before for cutting the DNA into smaller pieces. And then when you add calcium to this, you get that cutting of the DNA to smaller pieces, but only at locations where your target of interest was. So here, the histone mark or a transcription factor or something. Through doing this, you're going to have these small pieces of DNA wrapped around your target that are going to come out of the cell or nucleus that you're working with. And by using these magnetic beads to keep the cells down, you can take this supernatant, which will only have the DNA from the target region. So by doing it this way, instead of relying on immunoprecipitation, cut and run has a much higher signal to noise ratio than ChIP-seq does. And we'll see some plots of that a bit later. Once you have the DNA, you then still purify it and add the sequencing adapters like before, and the rest of the sequencing and everything is similar. Except the one thing I should note, because you have a much high signal to noise ratio, you usually need about 10 times less reads for cut and run sequencing than ChIP-seq, so it's a lot cheaper at the sequencing end, and can allow for you to use a lot less cells as input as well. A very similar technology is called cut and tag sequencing. And for this, things work basically the same, where you still have these magnetic beads. Instead of your antibody being bound to the imminase, it's bound to this special protein called TN5. And what TN5 does is it cuts the DNA, but also adds on sequencing adapters in a process called tagmentation, which is why it's called cut and tag. So instead of getting just those pieces of DNA, you get pieces of DNA that already have sequencing adapters. So reason I put this X here, you've basically skipped the next step. You already have your sequencing adapters in the actual sample. So then you still purify the DNA and sequence it. This technique works better for certain targets than cut and run does. In general, cut and run is usually a bit easier to get working. Cut and tag can work better for less cells and can also be adapted for things like single cell versions of this, which people are doing now. So it cuts the DNA and adds the sequencing adapter at the same time.
And that's what you don't use.
Yes. Yeah, I think they do both cut and run and cut and tag, but yes. And then a lot of the other like newer technologies are with tagmentation in general. And one big advantage of cut and tag again is like it has a single cell ability. You can't really do cut and run in a single cell way because you have the DNA coming out of the cells during the process. This you can do in like a single cell droplet. So it's, I guess I'll get to the next one. The similar technology is a DNAseq, which you might have heard of, which also has a single cell version. So because single cell DNAseq was already developed, you basically use the exact same kind of technology for single cell cut and tag. So the way a DNAseq works, which is a bit different, this is instead of looking for a specific target, is only looking for accessible regions of the genome. Here you just add the TN5 enzyme by itself. So it's not going to bind anywhere in particular, but it's only going to be able to bind to DNA that is accessible. So this part of the DNA here at the beginning, where the histones are very close to each other, is sort of like a closed chromatin state. And then you have these regions up here where the DNA is more open. So the TN5 just by physics is only going to be able to reach regions that are accessible, and it will do the tagmentation there, and you'll get these little pieces of DNA with sequencing adapters that you can then isolate and then go forward with that sequencing. So uh, the ChIP-seq, cut and run, and cut and tag are all giving you information on a specific target based on an antibody, whereas a tag-seq is just telling you about accessibility. One important kind of note is that things need to be somewhat accessible for any of these methods to work. So they have some trouble. You can do some of these for inaccessible regions, but if they're kind of super condensed chromatin, it can become difficult even to get those to work, even if you're interested in like a repressive mark or something like that. So you do need some level of accessibility for kind of any of this to work well. All right. Um, are there any questions before I move on just the technologies?
Or did, you said that you could use nuclei?
Yes.
You could
So
offer different methods.
yeah, so each is a bit different. ChIP-seq is probably where there's the most kind of possibilities to do it differently because you can just isolate chromatin individually, and yeah, different protocols are going to be quite different. One that's really nice is called Mint-chip, which is a multiplex version of ChIP-seq. It standardizes a lot of this by using barcoding of both the sample and the antibody. So for that, because you barcoded the samples, which is adding like a short nucleotide that's specific for each sample, before you do the chromatin immunoprecipitation, that makes it more standardized because you can do the immunoprecipitation for all the samples in the same tube. Um,
Some salt concentrations from
right.
certain bottles.
Yeah, and yeah, so usually like these are all going to be some sort of kit that is providing like the buffers and things. But yeah, the actual sort of chemistry of the different buffers is going to change a lot. So one big difference is for cut and tag, you need a much higher salt concentrations for the TN5 tagmentation to work. Cut and run doesn't matter as much, so that's one um, thing that usually makes cut and run a bit easier. It's like more forgiving of the conditions, whereas tagmentation is much more restrictive of what conditions it will actually work in. Another thing that's for the cut and run and cut and tag, since you're using cells or nuclei, the permeabilization of the cells and nuclei is really important because if you over permeabilize them, you're going to have DNA you don't want getting out. But if you under permeabilize them, the antibody's not going to get in and you're not going to get the target DNA out. So that can take a lot of troubleshooting as well. So yes, I think generally, as long as you're treating all of your samples the same, at least you can compare them to each other. But between different buffers or nuclei isolations or chromatin isolations, it will be quite a bit different for sure. The other thing that I didn't mention in kind of a detail, for chip sequencing, you're usually going to do some sort of cross-linking with PFA. So that keeps things a little bit easier to compare and a little bit more stable for these interactions. With cut and run and cut and tag, you're usually going to use unfixed, like fresh tissue. So that has kind of its pros and cons as well. And I guess just as another disclaimer, generally the antibodies and conditions that work for ChIP-seq are different than those that work for cut and run or cut and tag. So they're just they work quite a bit differently. So it's even though they give you similar data out, don't try to like put the two protocols together or anything. And for cut and run and cut and tag, one of the leading companies for um, those reagents is Epicipher that's here in Durham. So I highly recommend all of their stuff. There's also Active Motif, I think is in San Francisco, and then Cell Signaling also sells a cut and run and cut and tag kits. All right. So for the actual analysis part, I just wanted to throw this link out to start. This is from the bioinformatics core at Harvard. They have a lot of really great pages about sequencing analysis in general. I actually quoted a lot of them in the last lecture and forgot to actually say their name. So here's the link to their main page, but I'll show some of their figures and links later as well. Starting off with this one. So this is kind of their overview of at least what they say to do for ChIP-seq. I'll talk a bit about some of these parts and use a little bit different tools for some of them. But this is kind of the basic overview. So you're starting like we did last time with these FASTQ files, which are just the sequencing reads that you got from the machine, which just has that information of what nucleotides did we actually find, basically. You're then going to need to align those to the genome. Might have some sort of filtering. There's what we call peak calling, where we see where is there an abundance of these. There's basically QC steps all along here that I'm going to not talk about much. There's some different ways to compare which peaks you have between different conditions, which I'll talk about. And then I'm actually going to show visualization first because I think it makes sense to visualize the data really early on. They did it last for some reason. I'll talk a bit about motif discovery, and then talk a bit about annotation as well. So the first step is sequence alignment. So you wouldn't want to necessarily use the same aligners that we did for RNA sequencing. We talked a bit about STAR and how it's able to take into account that exons from RNA are going to be spliced, and that you then, if you align to the genome, there's going to be like a huge gap from the introns. With this, you don't have to worry because we took the DNA from the genome, so there shouldn't be any gaps really. So the main aligners that people use are BWA or Bowtie 2. Harvard had great figures for Bowtie 2, so that's what we'll focus on, but they work very similarly. And the basic idea, again, is that you have these sequencing reads, you have some sort of reference genome, and you want to map where the sequencing reads align. And this process will allow for some mismatches. So you can see here in red, a nucleotide is different in the sequencing read than the reference. And here in this dash represents that nucleotide was missing from that read. So depending on what parameters you set, you can kind of control how lenient it is, but generally sequencing isn't perfect, so we want to allow for a good deal of mismatches. If we were trying to sequence DNA for mutations or something like that, we would need a much higher read depth to accurately detect those. So we're basically going to assume that mutations we see are sequencing errors and not assume that they're genuine mutations in the DNA sequence. And that sort of lines up for kind of what we see here. We have several reads from this C, and we have one that says it's an A. So probably this, in this case, it says the human sample. Probably these cells didn't have an actual mutation there. This is just an error in the sequencing. Yeah. So one thing that you might need to do before alignment is to trim off those sequencing adapters that we added on. They're going to be usually included in those sequencing reads because that is something that the sequencer reads, even though it's not really important to us. So there's sort of different ways that these can align. The reference genomes are on the top. If you have the reads sort of already trimmed of those adapters, it will align kind of perfectly where you have no clipping is what it's called. So every part of the read aligned, and that's called an unclipped read, and that would be kind of the best alignment. Depending on what settings you give to Bowtie or to BWA, you can have this sort of hard clipping where it completely removes this part. So that would usually require you to tell it what the sequencing adapters are so that it can remove them, and then you'll have an alignment of the rest. If you, for some reason, didn't remove the sequencing adapters, you can also do this soft clipping where you tell Bowtie that there might be something on the edges that we want to leave. So if it finds a good alignment in the middle, but this little edge is not aligning, it will allow that and assume that that is some sort of sequencing adapter or extra DNA that's not of interest. So you kind of have to play around with those settings. In general, it's probably best to just go ahead and trim the sequencing adapters in the beginning. There is software that can do that, and then you don't have to kind of worry about this issue. But a lot of the newer aligners will still give you a pretty good alignment, even if you don't trim the sequencing adapters. So once you have the alignment completed, sort of the best thing to do is visualize it. I think this really helps you to know if things are working as expected. Like I mentioned for RNA-seq, I prefer using Integrative Genomics Viewer, or IGV, for this. This runs on your own computer and can open the aligned reads, which are called BAM files, directly. It will require an index, and if you don't have one, you can just use this tool called SAMtools to make one. It's quite simple. And it just needs those two to be in the same folder. But then you can just drag and drop them in there. Cut it off of my screenshot. But up in the top corner, there is a um, selection for what genome that you use for the alignment. And once you select that, when you drag and drop the sequences, it will know where you're at. I think, yeah, I had it on this one. So there's the genome there on the top, and then you can search for different regions of the genome or genes, and then they'll come up in the browser. For BAM files, it'll show you two sort of views. So one is this, what it calls the BAM file here, where you have the actual sequencing reads, and it's where they kind of lined up. So you can see these sort of stack over different regions, and the colors denote different mismatches between the reads and the reference. So you can see there's some, but in general, a good degree of alignment. And then on the top, you get what's called coverage, which we'll talk about a little later, but this is sort of how much of this like stacking up of reads you have along each part of the genome. And there's ways you can make files that have just that information. But this gives you a general sense of what your aligned reads look like. And you can see here I put the gene heaven. This histone modification is H3K4 trimethylation, which is a marker of promoters. And this is the promoter of heaven. So we can see that we have sort of a lot of this histone modification right here at the beginning of the gene, and there's not as much kind of on either side. So this gives us some confidence that we are measuring what we think we're measuring. So I think that's the first step I would recommend for these sort of data is if you know anything about your targets, like see if things are making sense. Particularly, usually H3K4 trimethylation is recommended as a control for a lot of these experiments because it has well-defined localization to gene promoters. So check, is H3K4 trimethyl at a gene promoter? Is it in genes that I would expect in my sample of interest? And is it absent from genes that I wouldn't expect? So for this case, we were sequencing astrocytes, of course, because I'm from Chala's lab. So astrocytes heavily express heaven or spark-like one. So we have a lot of it there. I didn't include it in the presentation here, but we also checked like genes like NUIN, which is that neuron marker, and there was an H3K4 trimethylation there. So that can help you get an idea of if things are working sort of as expected. Another key thing for most of these experiments, which I'm realizing is a bit small, is here on the top, I have H3K4 trimethylation again. For some reason, I switched to the gene neuroligin 2. But here on the bottom, I have an IgG control. So this is an antibody that's not going to recognize really anything in particular, and usually we'll have for cut and run or for ChIP-seq. And you can see here that there are some sequencing reads in this region for the IgG antibody, but they're not localized really to any particular location, and they don't seem to be accumulating at the promoter or anything. And they're much less intensity or coherence than we had for the histon mark. So this gives us some confidence that what we're seeing isn't just background. There are certain regions of the genome that are more accessible or are known to give you sort of a little bit of a peak with IgG. So you just want to have that to give yourself some confidence that it's not just that you are looking at a really accessible region and you would always see sequencing reads from there. Yeah.
Is there a metric to compare that, not just going gene by gene, but every year or something?
Yes. Yeah. Yeah, I'll get to that in a sec. Yes, perfect. So, yeah, I should have put maybe an explicit side about that. So, usually people will use H3K4 trimethylation as a positive control. It's helpful kind of at every step if you're doing ChIP-seq, cut and run, or cut and tag, because when you measure DNA, you should get some DNA from H3K4 trimethyl because the antibody is very good. So that helps you to know that it worked before you do the actual sequencing. It also is very highly localized to those promoters. So it gives you these sort of clean peaks like we're showing. Other transcription factors or histone modifications can be more kind of diffuse across the genome, particularly like the repressive histone marks are kind of everywhere. So they don't give you that really nice localization to the promoter. So it can be kind of hard to know if those worked correctly. Um, Having some sort of negative control, so either IgG or some people have also used histone 3, like just the total amount of histone 3 as a control. Um, uh, IgG is a bit more common, but histone 3 is what is used by that MinChip technique. In either case, having some sort of control that tells you what sort of the background sequencing is really helpful. Um, and then yeah, I have it on this slide. The other thing that's helpful to add is some sort of spike in normalization or for normalization. Um, so I guess I'll just explain normalization for a second. We talked about last time that for RNA-seq, we basically have the assumption that the total amount of RNA between samples is not changing. And so we normalized the libraries to each other based on how many reads we had. And we're basically only comparing, is gene A like 2% of the reads in one sample and then going to like 4% in the other sample is basically. Um, and that works pretty well for RNA, as long as you're not targeting something that, controls transcription or something like that. For DNA and this sort of chip-seq or cut and run, we would assume that there are cases where you're going to have drastic changes in the amount of histone modification that you're having or in the amount of transcription factor binding or something like that. So for this, we want to get a better sense of what the actual amount of DNA was that we put into sequencing. And the easiest way to do this is to add DNA from another species to the library before you sequence it. So this is called spike-in normalization. Usually people add E. coli DNA. As long as you're not studying E. coli, that works really well. And so um, I should have noted it on the plot, but you basically add the E. coli DNA usually during either this imminase activation or during the DNA purification, so that you have the same amount added to each sample. And then when you get sequencing reads, most of them will be for your species of interest, but a small percentage of them will be for E. coli. And then you use those reads here as what it calls reference reads. So this is from a package called QLS, which I'll get to a little later. But all of them do something fairly similar, where you can take the amount of reference reads between two samples and create this sort of batch multiplier that says, how much E. coli did each sample get and then normalize that way. So you still get a normalization factor for each library like we did for RNA-seq, but instead of coming from just the total reads, it's coming from this sort of corrected one that's corrected for the amount of DNA we put in. Um, so yeah, I guess does that mostly make sense? Um, yeah, so the ideal situation is that you isolate cells or nuclei, you have the same number of cells or nuclei, which can be kind of hard to generate. You add the spike in control, that's the same amount of DNA. You usually want that to be about 1% of your total sequencing reads, but that can take some troubleshooting because you probably don't know how many sequencing reads you're going to get, especially for a new target. So a lot of the kits come with some guidance, especially for the K4 trimethyl, but for example, if you're doing a transcription factor that no one's done cut and run on before, you kind of just have to guess and hope that you're right. You basically just want to get enough of the E. coli DNA for normalization, but you don't want to get so much of it that you're losing sequencing of the species you care about. Yeah, so that makes things most easy to compare between samples. It's still a bit hard to do those sort of comparisons because you rely so much on having the same amount of cells and protein in the beginning, but it's sort of the best way we have right now.
Or doesn't,
Yeah.
why is that? Why is there a -1 there?
Um, that is a great question for which I do not know the answer. Um, I'll get the link to Karis. They explain this in detail, and I don't know off the top of my head why it's got a minus one. I'm gonna completely sidestep it. Um, oh, the other thing that I haven't had much experience with, but was going to try to play around with for some newer, older data sets. A lot of people didn't do the spike in normalization originally, and particularly for cut and run and cut and tag, the Hinikoff lab that developed those techniques has since shown that there is some E. coli DNA in the TN5 or in the imminase, and that is enough to normalize the samples often. So I haven't actually tried that, like if you can add the spike in normalization, but they suggest that that is sort of a way that some older data sets could be better normalized when people hadn't had those controls. Because there's just, in the preparation of the enzyme, there is some left over because it is, I guess, a produced in bacteria, that there's enough residual. And because you add the same amount of enzyme to each sample, you kind of are doing the spike in, even if you didn't mean to. So I think that's a cool thing I haven't tried around with, played around with, but if you get enough reads of it, it could also be an option. So again, I think visualization is kind of the best place to start. What time it is. Okay, I'm gonna hurry up a little bit. Deep tools is one of my favorite for this kind of after you've done IGV. It's gonna make those like sort of genome-wide plots like Kira was alluding to, where you can see what's happening kind of at a larger level. Um, so it has a bunch of tools that are very useful, shown here. Some of them we won't talk about, but there's kind of a lot in that package. So, and they have a really good documentation on their website here. The first one, which I think is what Kira was specifically asking about, is called a fingerprint plot. This is useful for ChIP-seq, and I will show how it is somewhat useful for cut and run or cut and tag. So the idea is basically you have an input here in green, and you have your histone mark in blue, and you're plotting here what proportion of the DNA has what proportion of the reads. So if you have input, you basically assume that the reads are kind of relatively evenly distributed across the DNA. So there aren't particular regions of the genome that are getting a lot of reads, which is why you have something relatively close to um, like this diagonal. It's never going to be that close to the diagonal because some regions of the genome are relatively inaccessible just by their nature, but it's fairly close. When you do a histone mark that's highly localized, like H3K4 trimethyl, you get something closer to the bottom right corner, where a lot of the reads are at those gene promoters. So you have here, like they're showing this top sort of 3% of um, regions of the genome have like, what is it, 45% of the sequencing reads. So things are relatively concentrated to specific regions. You still have a lot of reads that aren't as concentrated, but it's fairly concentrated. And you can sort of measure the difference between your input and your target to show how kind of successful the immunoprecipitation was and how much of a localization you got from that. Um, and this will vary by different histone modifications or different targets for sure. They only show histone modifications here as examples, but transcription factors generally will be on the more diffuse side of this. And histone marks like K27 trimethylation, which is one of the main repressive marks, is just by its nature more diffuse. So you will see kind of less of this kind of specific localization. Um, this is not super helpful for cut and run. So here is showing ChIP-seq alongside cut and run. These ones are ChIP-seq and these ones are cut and run. These are all for a target, but I think it gets the point across that cut and run has a much higher signal to noise ratio. So you're only getting reads from those regions where you had the cutting and the antibody binding. So in general, you're very close to that bottom right corner, kind of closer than the best ChIP-seq would be. And sort of consequently, even if you compare a control to something like a transcription factor, you often see very little difference because even the control is only getting where the antibody bound, and it's still going to be a very low proportion of the genome that you're actually sequencing. So it's a little harder to interpret the fingerprint plots. Hopefully you get something like this where you can see the transcription factor is still a bit right shifted of the control, but often they'll be pretty much overlapping, and the fingerprint plots are not necessarily very useful for cut and run. The next tool from Deep Tools that you'll need to use is called multi-BAM summary. So this takes in multiple VAM files as input, and it makes this sort of coverage in what's called a compressed NumPy array. Deep Tools basically is using Python secretly because it's all through the command line, but that's the object it makes. And this can either give you some summaries of particular regions of the genome. So often people run this on all of the promoters for their species of interest, or you can do the whole genome. And one of the first things I like to do is plot PCA, which we talked a bit about last time, but is principal component analysis and gives you just sort of an overall view of how different samples are from each other. And so if you have, your different targets of interest, particularly at least the control and whatever you were interested in, you should see that those cluster separately from each other. So here we have the input kind of in the middle, H3K27 trimethylation, H3K9 trimethylation cluster together. Those are both repressive histone marks, so that sort of makes sense. And then H3K4 trimethyl and monomethyl cluster together because they are both marking sort of active promoters and enhancers, and have some overlap between each other. And generally are in more open regions of chromatin. So this can give you, again, another like early sense, did my experiment kind of work as expected? And then if you have sort of an experimental perturbation, you'd expect those samples to cluster with each other as well.
Listen, I use multi-BAMs, but I have, used different tools
Yeah.
after alignment to like determine how many reads are promoters or intramers.
Yeah.
Have you compared those? Do you know if they are, or is it not good?
I haven't really
for,
compared
like-
them directly. I like the multi-BAM summary because you can do it before you've like done anything.
Yeah.
So it gives you like a really quick view.
Right.
It's just whatever regions you tell it. So you'd have to give it a bed file. Yes. So a bed file is just like a file that contains different genomic regions. So usually for this multi-BAM summary, you would tell it, here is a list of all the promoters in the species I care about, and it will just compute for those. Yeah, it won't know that. Yeah, so it's only going to use what you give it. Potentially, yeah. 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 Basically you would use either the whole genome or something like the list of promoters from UCSC. Like it wouldn't be from the data set necessarily. So if there was like an alternative promoter in your data set, you wouldn't have that being shown.
Yeah.
I'm just like,
Yeah.
dumb question. What is the put map?
So that is like the IgG control.
Oh, the control path.
Or probably in this case for ChIP-seq, they might have done their control as without doing immunoprecipitation at all. Some people also do that. Just the DNA before immunoprecipitation. You can also do that way. It gives you basically the same information. But yeah, generally just one of those controls. The next thing to do is to make what's called a bigwig file. So this gives you that coverage that I was talking about, which I'll show visually in a second. Deep Tools has two versions. So one's called BAM coverage and one's called BAM compare. Sort of as the names imply, BAM coverage just does this with that sample by itself, and BAM compare is going to compare it to a control. So this can be really helpful if you have that IgG control where you have a lot of signal in it, particularly for ChIP-seq, and you want to say you got some reads pretty much everywhere for your control, But you just want to see where along the sample are my reads for my target much higher than the control. It'll sort of just divide along the whole each genomic bin to give you your coverage. So this can be really helpful to have like an early normalization of kind of each sample. Um, so again, just to visually show coverage, the reads look something like this, where they're just piled up, and the coverage is just sort of this score for each part. One big advantage is that this data is a lot smaller than the original data, because here we had every single read and what the sequence was and what the reference is supposed to be, and there's like a lot of information, also quality information on the sequencing. If you just cut it down to coverage, you just have at each genomic location some sort of score, usually between like 0 and 100. So this makes things very much smaller data-wise. It makes it a lot easier to kind of move forward. And these are often what you would show for like uploading to a database that you like, or a website, so that people can view your data. The bigwigs are a lot easier to show. And these are also, if you go to like any of the online genome browsers, this is what they'll show you. The next part for if you want to make like a heat map is called compute matrix and plot heat map. DeepTools breaks this up, which seems a little excessive, but is actually quite helpful. So basically, compute matrix does all the math that you need to make a heat map. So you probably like take that for granted, but there's a lot of kind of rescaling and like what is the intensity at each part of a heat map that needs to happen. So by doing this separately, it's kind of nice in that it saves that matrix file. And then any sort of aesthetic changes you want to make to the heat map, you can keep running off that same matrix. So if you need to change like the color map or something, or change the legend, you can rerun the plot heat map part without having to do all the computations again. So particularly if you're just trying to like make a plot look a bit better, it makes it a lot easier. So the way that I like to use this is in the reference point mode, which is essentially, again, you would have some sort of list of regions that you're interested in, usually gene promoters, and you would say line up all of these to something. So for gene promoters, usually we'll have the transcription start site of the gene in the middle, and then each row of this will be a different transcription start site. So yeah, like rows going top to bottom are different genes. And then left to right is along that gene's promoter. So you're lining them all up straight in the middle. So then if you have a histone mark, like, I don't think they even label what this is from the DeepTools page, but this looks like H3K4 trimethyl that's very concentrated at the transcription start site. You can see that like even across different genes, it lines up if you have them aligned to the transcription start site. And so if you can run those for different histone marks kind of in the same time. So here we have K27 trimethylation, and here we have K27 acetylation. So these are repressive and active marks respectively. Here they put the transcription start site on the left and the transcription like termination site on the right. I shouldn't use the example because I didn't talk about it. They also have a mode where you can give information like that, and it will kind of stretch or condense the gene based on that. So it's like proportional between those two, but not necessarily like linear, because some genes are bigger than others. But in any case, another cool feature is that it has k-means clustering as an option. So here they did a k-means clustering of 3. So this will cluster the genes by what this signal looks like. So you can see here this first cluster has a lot of H3K27 acetyl mark, or more of it than the K27 trimethyl. So doing something like that, you would find genes that were activated and not repressed. Conversely, these have a lot of trimethylation and not a lot of acetylation. Those are probably repressed and not activated. And then they had like a third cluster, which has a really high level of the methylation. So in doing this, you can find like sort of different categories of genes that might be of interest to you. And you can also do it for the same mark across conditions and things like that. Any questions on that part? So the key part is you're giving it those bigwig files, and you're giving it the regions that you want to look into. So this you can't really do in the whole genome, like you need to have some regions defined, particularly because it's going to try to align those. But you could make those your own. So I have done this before where um, we tried to identify enhancers from data and then plot on those enhancers. So even if they're regions you discovered from the data set itself, you can still use them for plotting. Um, yeah. All right. Cool. So after you've done your visualization, the next big step is peak calling. So this is sort of analogous to um, counting genes in RNA-seq, but for genes, in like RNA sequencing, it's already well defined where genes are. For this, it's less well defined. So there are three methods that I like, Max2, Homer, and then QLS, which is a way to actually run a bunch of different methods. But for this, since Harvard had pictures for Max2, we'll talk about that. So the basic idea is that when you did ChIP-seq or cut and run, you'll have sequencing reads that are oriented in one direction or the other based on which strand they're from. And the actual binding site of the thing you're interested in is going to be in the middle of those two. So it sort of assumes that there will be some sense reads and some antisense reads. There'll be some distance between these two peaks, and the thing you actually care about is in the middle. So it's trying to not find where the reads line up necessarily, but where the actual protein that gave rise to the reads is located. So it's a little bit different than simply finding the peak. And it varies a lot based on how you change the parameters. So here the reads are quite similar between these two. They're a little bit different, but you can see um, the antibody is supposed to show like where was the actual thing that you're interested in. So if you had something in the middle here, these sort of reads might make sense where you have some sense reads on this end and some antisense reads on this end. And they're all sort of around this central region. So maybe the actual thing you cared about was in the middle. But you can see if they're only slightly different or if even just the settings for the peak calling is different, a similar pattern could come from two nearby proteins. And you can see sort of here that even though these are only slightly shifted, you would get two different peaks. So Homer calls this focus peaks and unfocused peaks, but each of them have sort of different parameters. So what you end up getting is going to be kind of determined by sort of the assumptions you give the peak caller. Two things to sort of consider is one, like what the peaks of the target you have look like. So generally, H3K27 acetyl and most transcription factors have quite narrow peaks. So that's going to be like a very defined region. And most other histone modifications have relatively broad peaks. So they're going to be spread over more region of the genome and not really binding to one specific place. So here's an example with Homer where if you change the parameters, you get very different results, even though here on the bottom you have the same actual like data going into it. So one thing that needs to go into it is what was the actual length of your reads, and then some sort of estimate for that distance between the two. So you can see here on the top with the default parameters, you have a lot of small peaks detected because it's sort of assuming that you have those narrow peaks. Um, if you kind of change these, there's different modes in Homer between like fixed or variable length of peaks. You can see you get much different results. Here on the bottom, they have settings for like an assumption that these would be broad peaks, and you can see it's going to then group all of this signal together into one really big peak. Or if you have the settings for variable length, maybe these are actually, not so many little peaks, but many broader peaks. So unfortunately, there's not really like a right way to do this at the moment. It matters a bit like what your target is. And kind of, I would try to be as close to kind of what other people have done in the literature for your target, but um, there isn't really a ground truth of like what is the size of the peak supposed to be, particularly if you're doing like a new sample and a new target. So I think just have a little bit of knowledge that like what you put into this could change your results a bit. So that could be one reason why, one person might find a different result for the same target than you did. So it's a good idea to try analyzing the data again with your own peak calling method to see if that was causing some of the difference.
Localized on promoter regions.
What was that?
Stone marks you're looking at are. on
Um,
promoter?
H3K4 trimethyl is, and then H3K27 acetyl is, but there are like different ones for different regions.
Are there standard sizes? Millions?
Not really. Generally, the promoter marks are going to be like more narrow than the repressive marks, but there's not really like a very standardized size. Which is one reason why a lot of these are now adding sort of these abilities for variable length. So it tries to kind of infer what a good size is. So you can see that they're like recommended parameters for variable length peaks, I think is probably the best for this data set. Where it is getting that like this cluster here probably is one peak and not really two, but it does look rather like significant. And then this grouping, it's hard to say, is that two or is it like three, or one? So I think it's not well defined what is like the best answer, but just good to keep in mind that this will affect your downstream results.
Scholars too have a better argument. You can say it's a histone. You expect
Yes,
histone
so
peaks,
definitely, yeah. Homer has kind of different methods for different targets. So it will kind of take that into account for what it typically sees for those sort of targets. Um, um, Yeah, and so I didn't show it very graphically, but basically what it's doing is going along the genome, and it's going to put a tag where it's found that there are lots of sense and antisense reads kind of pointing at each other. And then it's going to measure how high is this amount of reads within that tag region, and is it much higher than the control sample? So if it is a lot higher than the control sample, we'd say that is a real peak. But for example, here there probably were sense and antisense reads pointing at each other, but this probably wasn't much higher than the control sample. So none of the different parameters actually called that a peak. And there are regions of the genome where you usually get some signal even in the control, which we probably would want to throw out. So it's not using like one set threshold across the whole genome. It's very local to different bins. And that is one of the variables that can change your result, is it'll use different bin sizes to decide like how big should it consider for each of the tags, or sort of how much of the local area to consider. I won't talk about Keras that much. The link is there. The main strength of this method is that since different peak callers give different results, they just made a method where it runs different peak callers. So it gives you this sort of output where it shows which peaks were found from different ones. And in particular, they focus on which peaks were found in all four of the peak callers that it uses. So they suggest just going with that and just trusting that peaks found by four separate methods are more likely to be real than the ones that were only picked up by one or two. But yeah, so I think that's a good way to approach it. Still, each of those have their own parameters, so it's not like you can, yeah. So it can become a lot, but it's a nice thing to try, I think, and gives you a little bit of robustness to how this works. The normalization that I showed was also from Keras. They have kind of a new one of their innovations is that For a lot of the peak callers, you had to normalize it differently for each one. They developed a normalized BAM file that allows like makes a file that can be accepted by all the different peak callers without having to normalize differently. So that's one useful thing they have as well. Once you've decided what the peaks are, you can generate a count matrix. So that's going to be rows would be each of your loci of interest or peaks, and then columns would be your different samples, and you'll get sort of a count for reads that are there. And then you can run differential expression between conditions like we did for RNA-seq. So I won't
Interesting.
go into detail.
So
Yeah.
you mentioned like you put a threshold to define the peak.
Yeah.
So, but is the peak binary or is it a
Usually, yeah, it's defined as binary. So for calling peaks, it's basically just trying to find which regions of the genome are important, kind of like what are the genes almost.
how do you not get a counts by counts?
So then you would run separate software with your peaks and your reads. And yeah, then basically count. Now that I know what regions are important, how many reads did I actually have that
I
lined up
see.
with those? Yeah. So you have to go back to the counts. Yeah. Yeah, so calling the peaks is basically finding what are the, like genes or what are the regions that actually matter in this data set. And then you go back and say, well, how much did I detect at each of those regions? And then you can do differential expression, like we talked about last time with, whichever your favorite tool is.
In that case, is that like for each peak you have a count?
Yes. Yeah. And I put the link here for diffbind. It's one that's a differential expression algorithm that's developed for ChIP-seq, but a lot of people also just use DNAseq2 or EdgeR or whatever they like for RNA-seq. It doesn't actually really make much difference. Going to what Chrissy said earlier, peak annotation is something that people are really interested in. So this is what are the different, like what do these genomic regions I found actually do? So this will be usually based on a database where there are annotations of where are the promoters, 5' UTR, 3' UTR, exons, introns, downstream are intergenic. The tool I like for this, because Christy suggested it, is called ChIP-seeker. It's in R and it does a bit better job than some of the others. A lot of the peak callers actually will give you some sort of annotation as well, but they're usually not quite as good as ChIP-seeker's annotation. Yeah, Homer doesn't seem great. Yeah, yeah, yeah. So yeah, this is basically just based on the literature again. So if your sample has something very different, these might not really be promoters, or whichever region. The other thing it's going to do is usually assign a gene, um, usually just the closest gene. So if you're setting a promoter, probably a good thing to do. If you're mainly focused on intergenic enhancers, probably a terrible thing to do. So it can vary like how useful that is. A lot of people will then do gene ontology analysis on the genes nearby the regions that they're interested in, or like where a transcription factor binds or something. So that can be kind of a varying utility. Some of those could be promoters that are actually driving those genes of interest. Some of them could be like proximal enhancers that are controlling those genes, but you could also get a lot of loci that aren't actually related to their closest gene. So you can kind of take those analyses a bit with a grain of salt. But one thing that you could do with like ChIP-seeker or something is find the promoters and then only do gene ontology on promoters that you identified, which probably are more likely to be related to their nearest gene. So different options there. A lot of people also, I should have put an example, but they'll put a plot just of the makeup of different genomic regions, for example, bound by a transcription factor. Is it binding to a lot of promoters? Is it binding to a lot of enhancers? Things like that, that could be of interest. And those things can also change across different conditions.
So what kind of data do you use to confirm your, the results?
So where, like what type of genomic region it is, or you mean just in general for it?
Okay.
There isn't a great way. So I guess it depends a bit on your question. If you have a total change in the histone modification, you could do a Western blot, or in the transcription factor binding. So you can do chromatin immunoprecipitation and then do a Western blot for sort of how much chromatin is bound. ChIP-seq is also compatible with like qPCR, so you can do the immunoprecipitation and then do like a specific target for PCR. So those are things that people have used. However, the cut and run sequencing in particular is sort of more accurate than any of those methods. And often you will not see a protein level abundance change in most conditions. So there isn't a great way to confirm a lot of at least the actual sequencing data itself. Um, there, yeah, there's some things you can try. So one is what some people will do is like an in situ hybridization for the region of interest. And then, for example, if you're mainly focused on like I discovered this region, is it actually an enhancer or something like that? You can do in situ hybridization of it and the promoter you think it's binding. And in a certain condition, if those are closer to each other, it likely means it's acting as an enhancer. There are assays where you can clone that putative enhancer or promoter region into reporter like plasmid and then transfect cells with that. So then you can see like if this region was an enhancer, does it increase transcription of something like GFP? And those can be done in your cell type of interest. So there are some things you can do for particular questions, but overall, it's kind of hard to like validate the sequencing itself. For like yeah, so there's a lot of method. Yeah, so you can do sort of a large scale screen of enhancers and things like that. So those can be done, yeah, like in Drosophila or yeast. StarSeq is another option where you would clone those different enhancer regions in front of GFP in a minimal promoter, and that can be done at scale. So you can have basically all the enhancers you identified from your sequencing. You can test in a different model. Of course, there you've taken the DNA out of its genomic context, so it might function differently. But that's something that people use quite a lot. And there are versions of that where you can even make it a virus and inject it into an animal. So you can get even in the native tissue what is going on. So there's some things, particularly for enhancers and promoters, but generally it can be kind of difficult. Of course, also for a histomodification, you can do, like we mentioned, Western blot or staining or things like that, but you probably won't have the resolution to really resolve how it's changing. Okay, cool. A little over, so I will go through this quickly, but the last thing I want to show is motif discovery. So this is finding if there's a particular sequence of DNA that's really enriched in one of your target regions. And so the main tools for this are called the meme suite. The sort of simplest one is called AIM, which is just using some sort of reference database. Do you have those sort of sequences in your sequences of interest? So for example, if you did a transcription factor and you search this database, does it have a known transcription factor binding motif? Because there's a lot of these that have been well studied. And these are what those motifs usually look like, where along position there are certain nucleotides that are much more common, which are shown as kind of like taller letters. The alternative, if you want to discover new motifs, is a tool called Stream, where it goes from the data and sees are there sequences that are more like represented or enriched in your data set. So for example, this is for the transcription factor ATF4. In this data set, it seemed like this sequence was heavily enriched in I believe this was ChIP-seq data. So this is a way you could find, for example, a transcription factor no one studied before if you wanted to discover de novo what its motifs are. Another thing that's really useful for this, you might think if you did a transcription factor, why bother if it's already known motif? You might see that there are other transcription factor motifs in your data that could suggest that other transcription factors are cooperating with the one that you're interested in. So it can still be kind of useful to do there. A final tool that's helpful is called TomTom, which lets you compare motifs to known motifs. So if you discovered motifs using Stream, you can then say how closely do they match with the database. and then kind of find are the ones you discovered matching with things that are known, or are they more novel? And that's actually what this original figure was taken from. So it gives you like a p-value for how close the two are. So you can see here on the top is the discovered motif from the data, and then this is the original like reference motif for that same transcription factor. So you can see they basically line up very well. You might have missed some of the nucleotides. For example, here, the first two and the last one that aren't very useful anyway are kind of cut off. So it can be slightly different, but it lets you see if the motifs match with what's known. Yeah.
Specifically feed those peak locations that you discovered, where it's like.
Yes. Yeah. So you would give it, you would give it the peaks that you're interested in, and then the some database. Or I guess if you're using Stream, it would only be the sequences, but for AIM or something, or the TomTom, you'd need to also have the database of known motifs as well. Which if you go to Meme Suite, well, it has the links there as well. Yeah, so that's like the very fast version. There's a lot of other things you can do with these data, but in general, aligning to the genome, visualizing I would do first. So I should have remade this graphic. You'll then call peaks and compare them between different conditions, and then do motif discovery and annotation. Yeah, and since we're already like 9 minutes over, I'll stop there.